Hello and welcome to the Meiosis practical, at least the introduction to it. So I've got a few short videos and hopefully my little picture in the corner won't obscure too many important details, but you never quite learn where this is going to happen when you're constructing your slides. But first of all, thank you for choosing the Meiosis practical. So I'll do a little bit of an introduction now, and then we'll go into the bits of the nitty-gritty. And then when you finally make it into the lab, you'll be prepped and ready for the mysteries of meiosis that await. I'm not going to lie to you that this will be a taxing practical, not because it's particularly difficult to actually create the preparations, but because it is a matter of numbers before you get preparations with the chromosomes at a state that they're actually useful. So it might take a lot of effort. You may need to make slides again and again and again, but do not be discouraged, because when it works, there's a big payoff in that you can get lots of data from a single slide. In fact, while I was preparing for this, I did a locust squash and I took over 200 pictures of stages of meiosis on the microscope. So when it works, it's brilliant. Do not get discouraged. So what we're doing is looking at meiosis, that peculiar division, which I shall go into in more detail, because I want to give you a little bit of background just in case your meiosis lectures are a dim and forgotten memory. Meiosis has been around for as long as the eukaryotes, or nearly as long. We don't really know. These things don't fossilize. But it has driven variation in eukaryotic cells since they first emerged. So while we study meiosis, we get a look at chromosomes and their evolution, the dance that they do in order to go from diploid to haploid gametes. It might be of interest because of meiotic drive. So there are some organisms in which you may wish to do some determination, and you can use meiotic drive as a way of pushing selected genes favorably into the next generation. And of course, in humans, there are a lot of mutations that are linked to problems in meiosis, particularly aneuploidy, where one chromosome segregates to the wrong pole of the cell when it's dividing. So one is +1 and one cell is -1. So that can be a problem. Polyploidy, lethal for humans, but really important in plant breeding, where you have a duplication of the whole genome, and that causes problems during meiosis as well, but plants are well equipped to sort through it and grow, and you get lots of polyploidy events which give rise to new races and perhaps eventually new species. And then there's the effect that genetic manipulation has on meiosis. So you are introducing a new gene. It might insert in one of the chromatids of the chromosome and not the other. And so there's an issue about error checking. And there has been some evidence that the inserted genes can have an issue with the points at which chromosomes form those double-strand breaks which heal the chiasma. And we'll talk about those a bit more later, because actually that's what these break and healing points are the ultimate aim of this practical. Why locusts? It's down to the genome size. A diploid human cell has 3 gigabases of DNA in 46 chromosomes, that's 23 pairs. The desert locust No, the migratory locus, which we're using, Schistocerca gregaria, has nearly 3 times as much DNA in only 22 chromosomes, 11 pairs. So when they pair up, when they join up during meiosis, you get these enormous, stonking great chromosomes which make life much easier. They are so huge that when you've got your eye in, when you've got not the eye of faith, but the eye of experience, you can actually locate them at the lowest magnification of your microscope, the ×4. For humans, once you've got the eye of experience, you need to spot them at least on the ×10 or the ×40. So here's an example of them. Locust chromosomes versus human chromosomes. They've been stained differently and photographed differently, so don't worry about the colour. That's an artifact of the staining. We could use plants, but that requires microdissection of very immature buds to get the pollen mother cells, so the cells that are undergoing meiosis, in order to get the right stages that you can see. The nice thing about plant meiosis is that it's synchronous, so all the pollen mother cells are at the same stage, but hitting the magic minute anther is the trick, and that doesn't always work. The biggest chromosomes in the plant kingdom tend to be in those plants that make bulbs, like onions and fritillaries and lilies and so on. And indeed, the middle section of this group of chromosomes is the snake's head fritillary, Fritillaria meleagrus. Those are just giant sea serpents of chromosomes compared to the others. And Phailcress arabidopsis, the white mouse of the plant world, is visible at 12 o'clock in this image. So they're quite hard to find, and you only get a single stage in the cycle, whereas in locust, as we'll see, we get the full extent of meiosis. Just to remind you, mitosis is the equational cell division, which means you end up with 2 cells at the end which are identical to the one you started at. And you'll be familiar with the stages of the cell cycle, the prophase, prometaphase, metaphase, anaphase, and telophase. They might have a few interruptions. There's a growth period after telophase before the cell is ready to divide again. The key in mitosis is that you start off with a cell, you end up with 2 cells that are identical. There is no mixing and assortment of genes and alleles within mitosis. The key differences in meiosis are many. So it's not a cycle, so the end is different from the beginning. There is a second division. So you have the DNA replication. The first division separates that into what would be like a diploid cell. It then undergoes a second division, so that you end up with 4 haploid products. So the end of the point of meiosis is 4 haploid cells, not 2 diploid ones. There is homologous pairing in meiosis, where the chromosomes from both parents find each other and align. And those homologous chromosomes swap sections via a tightly controlled process called crossing over. And crossing over has a physical aspect to it, which is what we're looking for, which is the development of chiasma. The chromosomes then independently segregate, so you have the crossed over bits and the maternal and the paternal bits segregating independently. That's called independent assortment. And the crossing over and the independent assortment lead to recombination. So the 4 haploid cells are all different from one another because their alleles have now been swapped over between the maternal and the paternal chromosomes. That's why they're not similar. And of course, they need to fuse with another haploid cell, which is fertilization or syngamy. But it's important to note that some organisms have a haploid and a diploid phase, like some of the seaweeds. So this is, I just took this diagram from one of the textbooks that is in common usage at the university. And here we have the meiotic process. It's not a cycle because they don't go round it again. You start with a cell at interphase. It goes through to prophase 1, metaphase 1, anaphase 1, telophase 1. And then you undergo the second division, prophase 2, metaphase 2, anaphase 2, telophase 2, until you get to those 4 haploid products, all of which, as you can see beautifully illustrated in this diagram, have subtly different chromosomes because of the independent assortment and the crossing over, which is seen in that little bubble labeled prophase 1. So there it is, prophase 1, which shows these beautifully aligned chromosomes, which have swapped over some arms at the chiasmata, or chiasma, showing the crossing over. So why don't they show meiosis photos in textbooks? The answer is because they look an absolute mess. You see this beautiful diagram with crisp, well-formed chromosomes. This is what they look like in early prophase. And they don't get clearer until much later. And then they move into metaphase 1. So we've got these 5 components of prophase 1. Those are the components we're looking at. Actually, we're only looking for one of those components of prophase 1. So we end up with things looking like a plate of spaghetti. And that's what we're going to be sorting through in this practical.